The goal of transformation is to cause bacteria, in this case E. coli, to pick up DNA plasmids from their environment. The bacteria are negatively charged because of the structure of their lipid bilayer cell membrane. And the plasmids are negatively charged as well because of their DNA's phosphate backbone. Since they are both negatively charged, they tend to repel one another. To facilitate this interaction, calcium chloride is mixed in with the bacterial cells. Once in solution, the positively charged calcium ions bind to the negatively charged phosphate groups on the cell surface and neutralize their charge. Plasmids containing the gene of interest, RFP, are then mixed with the treated cells at 4 degrees Celsius. The temperature is quickly increased to 42 degrees Celsius, and then reduced back to 4 degrees Celsius. This heat shock results in the formation of adhesion zones, creating a leaky plasma membrane. This may enable the cell to take up a plasmid from the solution. After heat shock, the bacterial cells are placed at 37 degrees Celsius and allowed to recover before being plated. Bacterial samples are now put on agar plates to test for successful transformations. The plate on the left contains the antibiotic ampicillin alone, and the plate on the right contains ampicillin and the sugar arabinose. On the left is a negative control plate, P-, where the transformation reaction was done without any plasmid. In the middle is another negative control, where transformation was carried out with plasmids, but where the gene of interest will not be expressed since the growth medium lacks the arabinose inducer. On the right is the permissive growth condition with arabinose that will let us identify successful transformations. After a 24-hour incubation period, any cell that was transformed with plasmid survives and divides to form a colony in the presence of the antibiotic ampicillin. However, only cells that were transformed with plasmid and grown in the presence of ampicillin and arabinose will result in red colonies. We expect these red colonies to have synthesized the mutant fluorescent protein, MFP.
Expression vectors. So the vectors that yield the protein product of the gene, or in simple words, the vectors which have the ability to synthesize protein from the inserted gene. Those vectors are expression vectors. We already know about the plasmid vectors, and these expression vectors are nothing but variants of plasmid vectors. These vectors are produced as a result of certain modifications and manipulations in the basic design of a plasmid vector. When such type of vector is engineered, it would look like this. So let's know about the different components and the elements which make this vector. First is of course the ORI, or the origin of replication, the sequences that allow their autonomous replication within the cell. Then we have the selectable marker, which helps in the identification of the transformed cells. These can be any of the being listed. Next element is the MCS, or multiple cloning site, also called as polylinker. It's a short segment of DNA which contains many restriction sites. The purpose of a MCS is that it allows a piece of DNA to be inserted into that region. Then coming up the most important element, the transcriptional promoter. So what is a promoter? It's a region of DNA that controls the transcription of a particular gene. Now these are the qualities of a good promoter. It must have minimal basal expression level. That means tight regulation of promoter should be there. The promoter should be easily induced, meaning that protein synthesis is only initiated when required by the introduction of an inducer such as IPTG. Then there are other elements necessary for gene expression such as RBS, or ribosomal binding site, so that protein synthesis can be initiated. Next is the transcription termination sequence, which would mark the end of a gene during transcription. Now there is an important difference you should know. There are differences in the machinery for the protein synthesis between prokaryotes and eukaryotes. Therefore, the expression vectors must have the elements for expression that are appropriate for the chosen host, either prokaryote or eukaryote. For example, prokaryotic expression vectors would have a shiny organo sequence at its translation initiation site for the binding of ribosomes. On the other hand, eukaryotic expression vectors would contain the Kozak consensus sequence. After the expression of the gene product, it may be necessary to purify the expressed protein. However, separating the protein of interest from the great majority of proteins of the host cell can be a difficult process. To make things easier, a purification tag may be added to the clone gene. This could be any of the listed ones. That's all.